I mean, okay, it's one day thing. Oh, I'm so sorry. Say, nah. I would just say, don't worry. I'm pretty excited. I just said it's just one day. It's too much stress for one day. It just goes after the presentation. You can take it.
Hello everyone, my name is Vimeth and this is my teammate friend. Uh, so today we are going to present advancements in genomic distance estimation. So no. Oh, all right. So have you ever wondered like how closely cat uh, cat and tiger, or how different they are? Like, so in here, like the different between such different um, organisms can be um, investigated like through the comparison of the genome. So the genomic distance is a popular measure that can be used to make these comparisons. Like, uh, so here, like genomic distance is a quantitative measure of the dissimilarity of dissimilarity or divergence between two genomes or DNA sequences. And so this used in bioinformatics, um, evolutionary biology, and comparative genomics to assess how closely related two genetic sequences are. So this different method can be used to compute genomic distance depending on the context and the type of analysis being performed. And and as here, like there are application of genomic distances estimation. um, for that I will give you like each of these, like a base idea of each of these. So first is population genetics. So this studying genetic diversity within species. And here we got phylogenetics. So this building evolutionary trees based on sequence divergence. And here we got evolutionary biology. So this identify horizontal gene transfer. And here we got metagenomics. So this comparing like microbial communities and species that identification. And we got here pathogen surveillance. So this, or we can say disease genomics. So this identify um, mutation linked to diseases and track viral or bacteria outbreaks. And cancer genomics. So this compare tumor versus normal genomes to identify driving mutations. And finally, we got here functional genomic. So this detect non-coding elements that across species. Okay, for this genomic distance estimation method can be divided into two types. One is alignment dependent methods, and alignment free methods. So in alignment dependent methods, it's like focus on determining the average nucleotide identity, as known as ANI. So it's through DNA like alignment. So this give us a formula like this. It says like ANI equals to total identical um, aligned bases divided by total aligned bases multiplied by 100%. So basically this ANI is a robust measure of overall similarity within homologous regions of two genomes. And here, so in here, in next slide, we are going to talk about more about our text clustering algorithms in next. And so these alignment free methods, this is like more efficient approach for handling large scale genomic data. And this is inspired by like text clustering algorithms and compress large genomes into more manageable sketches. And this calculate Jacquard similarity index using generator sketches. So we are going to talk about this later on. And okay, so minimash algorithm. So this is basically a popular what we it's like a popular algorithm in computer science field. And so in this technique, it's used estimating the similarity between two sets. So this was first introduced in information retrieval to efficiently evaluate the similarity between text. So this hashes the elements of the sets and then take the minimum hash value as a representation of the sets. And
You have in the bigger set, the A
Yeah,
and B,
yeah.
are sequences.
Yeah, these are two sets of sequences.
Okay. Then you
Yeah.
hash each sequence separately.
Yeah.
Okay. Then you take the minimum out of all of them or out of every group?
No, out of only these two groups.
Yeah, but this H minimum.
Yeah. Oh, yeah, this is separate.
Minimum of what?
Actually, this is the mini-hash algorithm, which is computer science in general. So in the algorithm, they do this separately. They transmission to compare this.
Okay, but the value itself, the H minimum, minimum of what? So you have how many value, and then you take the
minimum of the hash values that came from A.
all of them. Just take one value.
Yeah, it's all of it. Yeah. All right. So here is the Jacquard index. So so this statics used for measuring the similarity of two sets. So it also called as the intersection over union. So this give us this function. And okay, so in here, when A and B are disjoint, so it will show us here 0 because there are no common elements. And when A and B are the same, so it's giving us 1. So when there is no hash collision in the min hash values we got from set A and B, and it derives this particular function. So in other words, like we can use Jacquard index. Index is like to measure the similarity between two minimum hash values without finding the same finding the minimum value. and the importance of this JCAD index is that it can be obtained in fewer calculation steps and provides a practical approximation of ANI. So yeah, and go to the next slide. So here we got like a general algorithm. This is a simple framework right here, and we got genome A and genome B. So in here, first we do is KMR extraction, and this is the extraction, and it goes to sketch of each of these, and this will give us the distance measure of
What is
these. Sketch is more like or like it's like
It's a representation of the original set A. Like it's like in here we have reduced the dimension of the sequence into maybe one value or maybe a couple of values depending on the tool.
That's a mixer.
uh,
Then how you measure the distance?
so this will it's first extraction the KMR, and it will goes as hash function to sketch, and this will all generate distant measure.
Okay, so you use the sketches to measure the distance. What exactly you do with the sketches? The exact matching?
It's like the
I saw the G-card index, for
yeah,
example.
yeah.
That's
You know,
It measures the similarity, and using the similarity we can derive the distance. So it's one of the measures that we can use here.
yeah, so for example, you can take the view and
Yeah,
that view
how similar.
fraction. How similar. Is that true? It's your stuff.
I get lost. So I do.
No, not yours.
Oh, yeah, my bad.
He's a mathematician here.
You say so.
All right, yeah.
Yeah.
Go.
so the motivation, so our the main purpose is to review the advancements in alignment free genomic distance estimation techniques using popular state of the art tools. So and the main focus is that these methods, like based on their algorithms, accuracy, computational efficiency and scalability. So for our for our review paper, we got these research articles. So we got MASH. So this was published in 2016, and this is bin dash. It was published in 2018, and dashing it's 2019, and this is the fourth one we got, DNA 2-bit. This was published like recently 2024, and as you can see here, like this all tool were developed developed using different version of C++. Yeah,
By the way, the dashing paper, see the last author.
yeah,
Management.
yeah,
So if you go to his YouTube channel, you see a lot of
yeah. We will.
data structure, how you I would recommend his Right.
For now on, my colleague will continue.
So now we are going to look at the algorithms behind these four tools. And first we are going to look at MASH. So to give a general overview of the general algorithm that we explained earlier, we said we are going to extract the k-mesh, then generate the sketch, and using the sketch, we are going to take a distance measure. But in here, so what happens is, earlier we only had the k-mesh size. Now we have an additional parameter called the sketch size. So earlier we only took the minimum of the hash values, but instead we are now going to take couple of hash values at the very end of the sorted list. We are, in this case, S number of hash values. And when MASH was introduced, it was considered as one of the revolutionary publications. Why? Because it's the first this genomic distance estimation method that used mean hash, which was used in text clustering. And because of that, they derived their name MASH by removing this INH from mean hash. So we extract the sequences, as I mentioned earlier, they are going to extract the minimum number of hash values. In this method, the order of the expected runtime is given here. Usually n is the length of the sequence and S is the sketch size. Since n is really, if n is really huge compared to S, this order can be considered as linear. And once you have the and one more important thing in that tool, they have made available the functionality to use raw sequencing data. They used something called Bloom filter to filter the erroneous schemas. And they used Jaccard index first to do the distance measurement. Instead of directly using the union and intersection between A and B, now they are using the sketches. So almost similar. Here they are considering this expression is something similar to the intersection of A and B, and this is the union of the sketch A and B. And this is a similarity measure, but we need the distance. So 1 minus the similarity gives you the distance.
So intersection over union to
A
if
similar.
1 over 1 with the
Sorry, can you say?
1 over 1. So it cannot be, right? So they are exactly the same. What will happen?
That means then it's going to be 1. Because then the union, they overlap.
Well, what we want, two sets are exactly the same. Intersection over union. That won't be 1. Because the section would be smaller.
If A and B are the same,
Yeah, they are the same.
they overlap.
Intersection will be one of them, right? Size of one of them. They are same size. Union will be both of them. Size of them.
Yeah, if A and B
Not
are.
A and
Oh,
if they are exactly the same, then it will be 1. Then 1 minus 1 is 0. The distance of 0. Okay.
but since here the size is in the outside of the intersection, it's going to be the same.
Right. Yeah.
So this is the Jaccard distance they define. Then they say, let's assume the probability of having a single substitution in your sequence is D. then the expected number of mutations in a k-mer is k times d. Then they said we can assume that the number of mutations is going to be distributed according to the Poisson model. And using that, they calculated the probability of having no mutations. That means 1 minus probability of having a mutation, and you
So a
get this.
single mutation would affect KD. Why is that?
Because this is the single substitution. For each element have a probability, each mutation has, each substitution has a probability of D. Since there are k number of locations, you get the expected value by k times D.
So K is the size of the K.
Yes, this is the number of mutations per k.
There is one substitution in one genome, right? Probably we are considering all overlapping K. That's why it will affect.
I think this is not the probability of having single substitution per genome. It's per location.
Per location. Yes. Affected number of mutations. So you multiply that with the probability.
And using another method, they calculate the expected value of the probability, which is WOT. In here, W is the number of shared k-mers between A and B, and T is the total number of k-mers. Then we are going to compare this expected value and this expected value because we are focusing on D. And then is what happens when we equal WOT to this one. And from this one, we can isolate D and get this expression. Then after following couple of several assumptions, they got this value as the D and they call it mesh distance. Here J means the Jacquard distance that they calculated here. So that's the overall explanation of the mesh algorithm. Now we can move on to Bindash. I would like to complain a bit about Bindash. They only had 2 pages in their paper, so it was not explaining well.
It's probably
Yeah,
application of
previous tools, yeah. So it's an improved version of Mesh, and it is optimized for multi-threading. And in this tool, they are using a method called B-bit1 permutation raw min hash instead of the min hash that they used in the previous method, with optimal desensification techniques for better performance. Let's look at the algorithm. To generate the sketch, this b-bit min hash applies a multiple predefined hash functions. Earlier, we only used one hash function. This used a couple of them, and then select the minimum hash value using all the generated hash values. And from the minimum hash value, they are going to extract the lowest b bits, lowest b bits to construct the stretch. Then they are going to compare the, like let's say this is for the set A, then they are going to do the same thing for set B. Then they are going to use this pop count operation to compare these two sketches. And when that b-bit min hash become one permutation min hash, instead of using all the min hashes, they are using the most suitable hash function for a particular set. Then follow the same procedure. After that, they partition the hash values into buckets of size p. In each bucket, they extract the lowest b bits of the smallest hash value. So you have capital B number of buckets, and from each bucket you are going to extract b bits. So altogether there are b times b, capital B times simple b times bits. So they said in general.
How they decide on where to place the K, these buckets?
That's what's explained here. If V is the hash value that you generated for k-mer, it's usually assigned to this position bucket.
I see.
So this is the position of that p-value compared to the maximum hash value. This is a hash value, and based on that, they have divided into B buckets.
Probably in the MSB or LSB, maybe MSB, most significant bit of the number.
Yeah, So, but the problem was there might be unbalanced buckets because maybe some buckets doesn't have anything. To solve this problem, they used desensification, which orders buckets according to their method. They haven't specifically mentioned what is the ordering technique. And then insert the empty buckets with the smallest hash value of the next non-empty bucket.
Merge it,
Yeah.
reduce them.
Yes. So
Makes sense.
that's how they made the sketch. So once they have made the sketch, they can compare the distance between those two sketches using this measure. This is slightly different from the previous distance measure. Here you have 1 plus, 1 plus. In the previous one, we had minus.
Why they call it rolling?
I think they didn't specifically mention why it's called rolling, but I think it has something to do with the ordering. And desensification came from the inserting empty buckets with the smallest hash value.
Maybe it's related to rolling hash. There's a term called rolling hash. I'm hashing overlapping k-mer. Think about first k-mer, second overlapping k-mer. Now differ only one character. Because I'm shifting by one character. All overlapping k-mers just exactly perfectly match except one character. So instead of recalculating the hash value from scratch for the next overlapping k-mer, just do something for the extra character. Then merge it with the previous hash value, and that's how I come up with a new hash value. That's why they call it rolling, because they keep rolling the hash value. The very first hash value I calculate, they keep reusing it for calculating the hash value next. Maybe it's related to that.
Probably. Now we can move on to the next method called dashing. And this paper. In dashing, they introduces hyper-log-log sketches for genomic distance estimation, and it designed for high efficiency in speed. So this is a figure I extracted from the paper. So let's see what is this hyper-log-log first. So first, what we suppose they have generated random hash values here. All of these are 8-bit numbers. Here they have generated a set of 4 hash values, 16 hash values, and 64 hash values. Then they are going to look at the leading zero count. So leading zero count means the number of zeros at the beginning of the number. So here you have 2 zeros, 3 zeros. And in this set, the maximum least zero count is 3. Similarly, here least zero count is 5. Here the least zero count is 6. So what they argue is the maximum least zero count is equal to the log 2 of the size plus 1. Okay. So here log 2 of the size of this set is, say, in this case it doesn't matter. It doesn't work, but in these two cases, it's 4 plus 1 is 5, 2 plus 1 is 3. So that's the motivation behind using the maximum least zero count. So in here, they are explaining how the hashing and generating something similar to the sketch works. So here you have the original sequences, you hash them, or maybe the k-mers, you hash them and get 8-bit numbers. And then they are going to consider the first p number of bits to identify the CPU register address. So here you have 110, so it's going to be stored in the register starting with register number 110. And then under that register, you are going to store the rest of bits. And then Considering those numbers stored in a particular register, you are going to calculate hyperlog log. What is hyperlog log? It's the maximum least zero count. Here you have 2, 3, and so on. Then, using this hyperlog log, they are going to estimate the cardinality of these registers. Cardinality means the size of the set. So, they count the least zero count, and based on that maximum least zero count, they are going to estimate the total size of that particular set. So that's what they have done here. Then using this cardinality, the cardinality estimate is given here. So using this cardinality estimate, they are going to take an overall estimate in this step B. Then to measure the distance, we know we need the cardinality of set, a measure for cardinality of set A, set B, their union, and their intersection. These two steps are quite straightforward, but in this method, finding the intersection is a bit hard. They said it's going to fail if you are going to use the same method for the intersection. Therefore, they used a different estimate called Earle's joint maximum likelihood, which I didn't understand that much. And this is the algorithm behind DNA 2-bit. This is a really good paper, a comprehensive one. In here, hash values of extracted k-mers are allocated to bins of fixed number L through the modulo operation. Modulo operation means you are going to divide by L, then the remainder count is going to decide your bin. And SIMHAsh function is used to represent k-mers within a specific bin. In general, minhash use the minimum value. SIMHAsh usually use something like a weighted average in general. But here, this is how they approach this method. For k-mers calculated hash, then they used a separate function called sign, where they check the sign bit of each k-mer, and if it's 1, they assign 1. If it's 0, they assign -1. And using these sign hash numbers, they generated a bit array for each node. So here we had L nodes. So here we are going to generate L number of bit arrays. This is the algorithm they used. And
So the 0 and 1 represent the entire hash value. One character.
here, yes, enter hash values. And using these bit array nodes, and then they measure the distance using this equation. Oh, then they compare these two bit arrays using the Hamming distance. So here they didn't go for the checkered index. And I think this is much more clear. So you have the k-mers here, and hash values. From the hash values, you have signed ones and unsigned ones, and you do the modulo operation. And based on modulo operation, these are going to divide it into different bins. And each number has a sign. Based on that sign, it will be either -1 or 1. Then you can calculate the bit array by adding these numbers. So you have one bit array for one hash set. So suppose this is coming from the sequence A, then you are going to something similar for sequence B. And then by applying the Hamming distance, you can calculate an estimation for the distance.
Then the hash function be specially designed because you need to have a balance bins. If all of them scattered around some of them, then it will
Yes.
fail.
Yes, that's a good point.
You know which hash function they use?
I remember they mentioned something like murmur 3 is not going to work, but I don't exactly remember which was working. So let's see the simulations. We used two
This is your own.
Yes, my simulations. So we used two reference genome sequences to compare the performance of these four tools. Does anyone remember what was in the first content slide? The figure on the first content slide. Do you remember what I was making last? Yes, there were two animals. It was a cat and a tiger. I asked how to measure the distance. So we are going to do that. So I extracted the reference genomes for these two species. And for the mesh tool, I use this GitHub repository. This is how I install the tool. And you can verify whether something is installed properly by running the help command. And they gave this. And these are the general commands that you can use to generate the sketches and measure the distance. In this case, you should generate the sketches first to calculate the distance. Oh no, in this function, you could generate the distance without calculate the distance without generating sketches separately. They had that functionality. So this was the distance measure that they generated. Here it means they had 437 matching k-mers out of 1000. And for bin dash, quite similar. This is the verification, and these are the commands. They didn't have the functionality to do the distance estimation without generating the sketches. And dashing gave me a hard time because the dashing, we have a Mac device, and this dashing, this GitHub repository, the tool given in here is not working well with Mac. Then I found out they have updated their tool with the name dashing 2 so that it will support Mac Silicon. And we executed that. And even to execute that, we had to do some changes in the make file and do the compilation. And these are the commands that we can use to do the sketching and distance calculation. And if I remember correctly, this tool can do the calculations in one, like do the distance calculation simply by inputting the FASTA files. And this is DNA 2-bit, quite similar. And let's look at the results. So we can see mesh taking a lot of time for the sketch generation. Actually, in all these tools, what takes time is the sketch generation. In mesh and dashing, as I mentioned earlier, it has the functionality to run both of these steps in one go. So I had this total line. So this is the time when the wall clock time when we combine these two operations in one go. So based on these, DNA 2-bit had the best performance. And maximum RAM usage was better in bin dash. Here for the DNA 2-bit, we got 1091. I don't know whether this is particularly for these two reference genomes, because I didn't compare this. I didn't check the maximum RAM usage for other species. And sketch file size is lesser in dashing, which was mentioned in literature. And these are the instructions per cycle. And distance measurements are different, but we can say these three are a little bit closer compared to the one calculated in dashing. If we compare the advantages and disadvantages, MASH has a high accuracy based on literature, and they generated large sketch files, and they are not going to work well for unbalanced sets. Suppose your sequences are not like quite different in size, it's not going to perform well. And in bin dash, it's faster than MASH, and it's going to work well with larger sets, but still the sketch sizes are large. They improved the speed, but they were not able to improve the sketch size. And in dashing, they produced the smallest sketch files, as we saw in the previous slide, but they had low accuracy, which was we saw in the previous slide. And DNA 2-bit is the fastest, but they are going to be slow when the two sequences are quite different from each other a lot.
You know, quite slow and
DNA 2-bit. Mm. No. Oh, I don't know why exactly it's.
maybe it's related to the number. They relies on value, the content of the hash
Yeah.
the sign. Too many of them. Content was actually carrying one by one value. That's my guess. Sure. Okay.
And in conclusion, these are the tools that we reviewed in this presentation. And with respect to the word clock time, DNA 2-bit showed the best performance. And also it provides versatility with different hash functions, which we didn't tried in our simulations. Overall, match showed the worst performance, but like worst performance with respect to the word clock time and memory allocations, but it was a breakthrough during its time. So actually the best method depends on the application, not just based on those parameters. Match the tool to the task that validate the result with the real world scenario. And these are the main references that we used. With that, I'm going to end my presentation. Thank you.
Any questions?
Questions?
I mean, I have one. Go back to.
Oh, the
So here, like,
yeah.
all of them have the same. Most.
For the first two.
Yeah.
For the first two. But here the sketch size is because actually in dashing, sketch file size is really good. Yeah.
Yeah.
Case size is a problem.
Yeah,
Or is
but
there any other one?
any other tools? Yes, there
There
are
are-
many other tools
yeah.
actually, and there are alignment based methods as well. These are just alignment free methods.
I really thought of out of curiosity. Something wrong.
That's a good question.
I think it heavily depends on what I mentioned in the conclusion. You have to select the tool depending on your application, what you are looking for. Like if it's reducing the sketch file size because you don't have enough space, then you should go for the smallest
How do
sketch
you
file.
define it?
I didn't have the chance to compare the accuracy because I couldn't find the ANI value, the ground truth value.
So this
The accuracy, the comments about accuracy are based on the literature.
okay. So someone would
Yeah.
evaluate
Yeah.
them.
Yeah.
Yes.
I was wondering, since you only have two species, and that they are similar or not. So not
Yeah.
detect in advance what is the distance
Yes.
or it grows, and then that will detect it.
Yes. There was an online tool where we can upload the FASTA and compare two genomes, but the
Yeah, you could put
upload.
the whole genome alive to it.
Yeah, but I couldn't upload the FASTA file because the FASTA files were on 2.5 GB, so I couldn't upload it.
Yeah. What's that? Advantage.
I think we
No.
can include another advantage here. Sometimes, based on the speed, it outperforms DNA 2-bit as well. So not just the sketch file size, it's faster as well. Mm.
But then the
Yes.
Yes, I think so as well.
so are there
I think when someone is limited with the memory space, it's like when someone is limited with their space in their device or the server. I think that's the only case that I think of right now. Or maybe like these are just a couple of advantages and disadvantages. Maybe we need to look more deeper into their pros and cons and decide.
like
Hmm.
I don't know.
I think there are problems
Mhm.
Your goal is just to know if they are similar or not. You don't care about how similar. And think about you want to set a threshold. Anything above 50%. They don't care how similar. One, like they don't. As long as above 50%. I'm good with that. Some of the tools tell you that it's working 30%. Filtering them out because they are below. In fact, they are above them. It could be not filtered out. But just slightly higher than. But because of the tool protected wrongly, but the slightly will be filtered out. This we call them false negatives. I measured, I evaluated that myself. In a sense, the measure itself, the value, it's very small, although it should be not in a sense to be filtered out. You don't do any filtering. But if you want to use it, this use case that I mentioned will result in detecting or similar.
Since it was mentioned yesterday, we tried to compare the same genome with itself,
Okay.
and all these tools predicted it correctly. Like
Okay.
they gave the similarity score as 1.
Yeah, for yesterday's discussion, it's alignment-based method, right? They align the genome to itself.
Mm.
So it's not like they'll detect it wrongly, but
Mm.
why it's 85% of similarity, although it's the exact same genome.
Yeah.
But that's a good point.
Yeah. And one more thing, I think this low accuracy problem comes in, like become be huge problem when they are so different, not when they are closely related.
So the distance will be minimal, although they are different.
Yeah.
Yeah, that will be false positive. The genomes are totally different, but the similarity
Mhm.
measure will tell you that it's very similar. Really nice that you quantified instruction.
Yeah.
Why you did that?
just because the values were there in the output file,
Okay.
so I wanted to play with them.
I like that. It is computer architecture. Any more questions? Right, thank you so much. Okay, we have two more presentations. We take a group picture now. Take a break. The picture. You're not allowed to leave. Too short. It'll be interesting one. Okay. Or you can order lunch and have it here. Yeah, let's take group picture. I'm not sure which background. Should we have it here? Yeah, this is tucking the face. We can have it here. Or maybe here. Here, yeah, it's not coming to my face, so it should be fine.